The escal photophore of Ceratioids, Pisces, Ceratioidei, a review of structure and function. The article deals with the structure and presumed functions of the escal photophore found in the bulbous tip of the cephalic fin array, or elysium, situated on the upper part of the head, in metamorphosed females in most species of deep-sea anglerfishes, ceratioids. The escal photophore consists of the light gland proper and certain accessory structures. An accessory structure common to all species is a light-proof cup enclosing the light gland and provided with a distal opening, while the escae and a number of ceratioids in addition possess reflecting structures, tubular modifications of which form light guides in some species. The light gland proper is a roughly oval or spherical body, typically consisting of radiating branched glandular tubules arranged around a central escal cavity, which communicates with the exterior via the vestibule, a usually slit-like epithelium-lined space lying above the distal part of the light gland. All lumina within the light gland contain bioluminescent symbiotic bacteria. The esca is generally thought to function as a lure, prey being attracted by the light emitted from the photophore and movements of the elysium. A possible additional function may be the ejection of a luminous material to confuse predators. The meso- and bathypelagic ceratioids, or deep-sea anglerfishes, constitute a suborder of the Telosian order Lophiformes. The larvae live mainly in the epipelagic zone and descend rapidly into deep water at the start of metamorphosis. The ceratioids are characterized by a pronounced sexual dimorphism, one of the striking differences between the two sexes being the presence of photophores in metamorphosed females of the overwhelming majority of the approximately 150 species and the total absence of luminous organs in the dwarfed males. Bertelsen, 1951. Several types of ceratioid photophores are known, the most common being the single light organ present in the female of most species. This photophore, which contains light-producing symbiotic bacteria, is almost invariably situated on the upper part of the head within the bulbous tip, the esca, of a generally elongated, modified dorsal fin ray, the cephalic fin ray or elysium, figure 1. A second cephalic ray fin, carrying a terminal bulb, is present just behind the elysium in juvenile deratids and larval ceratids, suggesting that a second bacterial light gland may be present in this so-called posterior esca in young and larval fishes. Furthermore, ceratid females possess additional bacterial light organs, caruncles, situated at the tips of modified dorsal fin rays in front of the normally developed dorsal fin. Bertelsen, 1951, figure 12. Special conditions are found in the genus Linophrine, the females of which have a dual luminescent system, vis-a-vis the bacterial photophore of the esca, and a barbel containing a number of small separate light organs with intrinsic intracellular luminescence. Hansen and Herring, 1977. Figure 1. Habitus of 25 mm young adolescent female, Melanocetus johnsoni. Figure 2. Diagram of median section of a ceratoid esca, rostral to the left. The figure emphasizes the continuity between the epidermis and the epithelium lining the interior lumina of the esca, the vestibule, and the escal light gland proper. The light gland proper is enclosed in a light-proof cup consisting of an inner reflecting and an outer pigmented layer. A duct connects the central cavity of the light gland with the vestibule, left arrowhead, which in its turn opens to the exterior via a short duct leading to the escal pore, right arrowhead, situated posteriorly on the distal surface of the escal bulb. Arrow points at blindly ending branched tubule, originating from the epilithium lining the walls of the vestibule. The distal appendage on the escal bulb contains a single light guide with a terminal window. The present article reviews the limited knowledge of the structure and function of the photophore of the alisial esca, henceforth termed esca, which is the best known of the ceratioid light organs. The article is based partly on published records, partly on unpublished observations. All lengths of specimens are standard lengths. The deep-sea anglerfishes are comparatively rare and generally dead or moribund when brought on deck. There are consequently only a few observations of the behavior of live fishes, and it is difficult to obtain either well-fixed material for histological studies or live specimens for physiological experiments. Structure of the esca. Of the more than 100 species of ceratioid possessing an escal-like gland, the escae of about 20 have been studied by light, LM, and electron, EM, microscopy. Table 1. The escae show an amazing interspecific variation in morphology, being provided with various types of characteristic appendages, some of which are known to function as light guides, and some of which are probably sensory. In the following account of escal structure, it will be assumed that the movable elysium has a vertically upward pointing position. With this orientation of the esca, the generally used terms anterior and posterior are identical with the terms rostral and caudal, respectively. Horizontal, frontal, and sagittal sections of the esca refer to sections cut with the plane of sectioning at right angles to the axis of the elysium, parallel to the axis of the elysium and at right angles to the median plane of the esca, and parallel to the median plane of the esca, respectively. Most drawings of the escae depict them as viewed from the left side with the elysium in the upward pointing position. In the subsequent sections dealing with escal morphology, specific citations for histological details have been largely omitted. The primary references are listed in Table 1. Escal photophore. The escal photophore consists of the light gland proper and certain accessory structures. Figure 2. The light gland proper is always partly enclosed within a light-proof cup, termed the cup in the following account. A usually slit-like epithelinium-lined space, the vestibule, lies above the distal part of the light gland, situated in the aperture of the cup. The vestibule is connected with the interior of the gland through one or several ducts, and a short duct from the vestibule opens to the exterior on the posterior distal surface of the esca, the escal pore. In some species, the light gland is associated with light guiding and reflecting structures, generally situated on the distal part of the esca. Except for a few small specimens, the lumina of the escal light glands of all metamorphosed females examined are filled with a granular content, identified as bacteria in electron micrographs. Bassett 1966, Hewlett and Musil 1968. Since the light is produced by the symbiotic bacteria, the light glands of larval and small metamorphosed specimens containing no or only very few bacteria are assumed to be non-functional. In what follows, the structure of the bacteria-filled escal light gland of metamorphosed specimens will be dealt with first. Table 1. Observations of escal luminescence and histological structure. Unmarked species names and sources indicate cases in which only the histological structure of the esca has been dealt with. A single asterisk marking the name of a species indicates that luminescence has been observed in live species. Double asterisks that the published records contain both observations of luminescence and histological structure. A single asterisk marking a source indicates that its text contains only observations of luminescence. Double asterisks that both luminescence and histological structure are dealt with. Okay, so it's a list of species and then a list of sources and the specific types within each of the 6 species. Cup of escal photophore. The light-proof cup enclosing the light gland proper always has a distal aperture through which light is emitted. Figure 2. Its base is attached to the distal end of the elysium, being in some cases fastened to a socket formed by an inverted cone of cartilage carried by the tip of the elysium. Figure 11. The cup, which consists of an inner reflecting layer backed by a relatively thin outer pigmented layer, is situated beneath the skin, being separated from the dermis by a layer of loose connective tissue. The cup's reflecting layer contains cells whose cytoplasm is filled with birefringent crystals, probably consisting of guanine. Its pigmented layer cells with melanin granules. Since most crystals are preserved in situ in the relatively thick LM sections, the crystal-containing cells are clearly identifiable when viewed by incident illumination or through crossed polarizers. In the thin EM sections, practically all of the membrane-bounded crystals have broken out of the plastic during sectioning, leaving a number of roughly rectangular holes, each of which has contained a crystal. Figure 3a. In specimens fixed in one of the conventional fixatives previously used for LM, such as formalin, and stored for a long time in ethanol, the birefringent crystals, originally present in the cup and other reflecting escal structures, are lost. Escal light gland with typical radial tubules. This type of light gland, built as a compound, branched, tubular gland, is characteristic of the majority of species examined, Figure 2. Published records on the detailed structure of such light glands are available for the following species: Melanocetus johnsoni, M. murrayi, Claniophryne draco, Onerodes escheridae, Haplophryne mollis, and Linophryne arbifera. These earlier records have been supplemented with a few